I am Dr. Saqib from my channel Learning Anatomy, and today I will discuss with you the structure of the cerebellum. Yes, and in that lecture, I will discuss with you the white matter, the gray matter, the various nuclei present in the cerebellum, and the various circuits made by these cells. These are known as the circuitry of the cerebellum. So cerebellum is made up of an outer covering of gray matter called the cortex and an inner white matter. Deep in the white matter of each hemisphere are three masses of gray matter constituting the intracerebellar nuclei. So there are four total nuclei. I will tell the detail how they are called four and how they are called three. Cerebellar cortex is a large sheet with folds called folia lying in coronal or transverse plane. You see this picture? Yes, this is the cerebellum, this structure, right? And this is, I already told you in my first lecture, right? This is the brain stem. And here you see, these are the folds called as the folia. These are the folds produced here as the folia. Each fold or folium comprises a core of white matter covered superficially by gray matter. So this is the folium, it's superficially covered by gray matter, and deep inside this is the white matter. So if a paramedian section made through cerebellum divides the folia at right angles and the cut surface has a branched appearance named as the arbor vitae. So here it is this is the branched appearance of the white matter. You see, these are like the branches of the tree, and this is called as the arbor vitae. In Latin it means tree of life. Arbor vitae means tree of life. Let me zoom. Here you see, this is a beautiful tree like appearance, right? So that is a arbor vitae. So let's move to the structure of the cerebellar cortex first. Cerebellar cortex. So there are five types of neurons present in the cerebellar cortex. These are the Purkinje cell, granule cell, stellate cell, basket cell, and the Golgi cell. Here you see all these five types. These are first of all, you see just these are the granule cell here, right? These are the granule cell. These are the Golgi cell. Here you see the Golgi cell. These are the stellate cell, right? And these are the Purkinje cell here. And these are the basket cells. This is the identification or spotting of the five types of the cells. And the gray matter of the cortex throughout its extent has a uniform structure, may be divided into three layers. It is divided into three layers. An external layer known as the molecular layer, a middle layer known as the Purkinje cell layer, and an internal layer which is known as the granular layer, right? Molecular, Purkinje, and the granular layer. And you can remember the sequence by a mnemonic that the outside is the M, P, G, and there is a mnemonic is the man, please go. So with M for man, molecular, and P for Purkinje, and go G for granular layer, right? You see the three layers here. Yes, you can identify these three layers. This is the cerebellar cortex. First layer is the molecular layer, right? And the second layer, this layer is the Purkinje cell layer. And this is the dotted line. This is tracing the layers over here. And this is the granular layer, right? These are the three layers of the cerebellar cortex. These are the three layers, the cerebellar cortex. Below is, of course, the white matter, below that. So let's discuss these layers one by one. The molecular layer. It contains two types of neurons, outer stellate cell and inner basket cell. So identify these. Let me zoom in for you. Yes, here it goes. Here you see first was there the stellate cell. Yes, you see this is the stellate cell, and the other is the basket cell. You see here. So this is the basket cell. Basket cell. This whole layer, this from here to here is the our molecular layer, right? So first of all, we can see this stellate cell in that, and the other one is the basket cell. These are the two types of the cell present in the outer molecular layer. These neurons are scattered among dendritic ramification and numerous slender axons that run parallel to the long axis of the folia. The series long axis of the folia is that follow the red laser, and they run parallel to that. Neuroglial cells are present between these structures. So the next layer, Purkinje cell layer, very important. These are the large Golgi type 1 neurons, right? Here you see Golgi type 1 because this is a flask shaped Purkinje cell layer, right? These are the neurons. You can see the laser, this. And they have a long axon. Here it goes. This is the axon. You see the laser. And these are the dendrites. So any cell, a neuronal cell, any neuron with a long axon is called as the Golgi type 1 neuron. So Purkinje cell is a Golgi type 1 neuron. It is flask shaped, I told you, arranged in a single layer. Yes, you see, this is a single layer. Arrangement is in a single layer. Here it is. This is the layer. In a plane transverse to the folium, dendrites of the Purkinje cells pass into the molecular layer where they branch profusely. Here you see them here, right? They are going branching profusely. The primary and secondary branches are smooth, and subsequent branches are covered by small, thick dendritic spines. These are the terminal branches covered by dendrites. The spines form synaptic contacts with the parallel fibers derived from granule cell axons, right? These are the granule cell axons. I will tell you later on. And you here, I think it is very appropriate to show the enlarged zoom. This is the Purkinje cell. This I told you is the axon. It's going down. Here it is. This is the axon. And these are the dendrites, right? And this is the axons, branching profusely. That's sorry, dendrites. These are the Purkinje cell dendrites which branch profusely. These go into this molecular layer. You see this beautiful orientation of the Purkinje cell. You see this is the cell body, which is this, right? This is the cell body of the Purkinje cell, and this forearm is its axon, right? Yes, here it is the axon, and these fingers are the digits or fingers. These are the dendrites. Here, these are the dendrites. This is a beautiful orientation to make a concept of the Purkinje cells. Structure of the cerebellar cortex continues, and in the Purkinje cells, at the base of the Purkinje cell, its axon originates and passes through granular layer and enters the white matter. Here you see, this is the axon originating. Here it goes, here it reaches here through the granule layer, and this enters the white matter. In the white matter, it gets myelinated, and it terminates by synapsing with cells of one of the intracerebellar nuclei. Like this is you see, these are the intracerebellar nuclei. You're going to learn very soon in this lecture. Purkinje cells are the only outflow from cerebellar cortex. This line is very important, that all the other fibers are coming as an inflow to the cerebellum, and the Purkinje cells are the only outflow from cerebellar cortex. Here they are going, right? So the granular layer, the third layer, last layer of the cortex, is packed with small cells with densely staining nuclei and minimal cytoplasm. You see, let's enlarge the picture to see the granule cells. This is the granular layer, and these are the granule cells. You see the labeling, these are the granular cell. They see it, these are. From each cell arise 4 or 5 dendrites, which make claw-like endings and have synaptic contact with mossy fiber input. Here you see. Here they are, making a claw-like dendrites, and they make synapse with the mossy fibers. These are the mossy fibers endings. The axon of each granule cell passes into the molecular layer where it bifurcates at a T junction. Here I enlarge it again. Here you see. These are the axon of the granular cell. Here they bifurcate. This is a T pattern, and they are running parallel to the long axis of the folia. So these are called parallel fibers and run at right angles to the dendritic processes of Purkinje cells. These are the Purkinje cell, and they are running parallel. These are the dots. These are showing the parallel running of these fibers, the granule cells. Majority of the parallel fibers make synapses with the spinous processes of the dendrites of the Purkinje cells scattered throughout granule layers of the Golgi cells. So let's now spot these Golgi cells. You see here, these are the Golgi cells. Yes, right. So they are scattered throughout the granular layer. These are the Golgi cells. The dendrites of Golgi cells ramify in the molecular layer, right? They ramify in the molecular layer here, and their axons terminate by splitting up into branches that synapse with the dendrites of the granular cells, and their dendrites synapse with the dendrites of the granule cells. You see this again, right? These are the axons, and they synapse with the granules of with the dendrites of the granule cells here. So few words now about the functional areas of the cerebellar cortex. Cerebellar cortex may be divided into three functional areas. Here these are, right? One, this area. This is the cortex of the vermis, influences the movements of the long axis of the body. Long axis of the body, including the neck, shoulders, thorax, abdomen, and hips. Here is shown the control of the neck. Here the shoulders, and then this thorax, abdomen, and the hips, right? So this whole thing is an unfolded schematic of cerebellum demonstrating body map areas, right? This is the thing. Body map areas are shown in that. Area lying immediately lateral to the vermis is called intermediate zone. Paravermis, this is the intermediate zone, and it controls the muscles of the distal parts of limbs, especially hands and feet, right? This whole thing is shown as a vermis, and this area, this is showing the control of the hands and feet. Lateral zone, right? What does that show? Of each cerebellar hemisphere. These are the lateral zones, cerebellar hemisphere. They are associated with the planning of sequential movements of the whole body and is involved with the conscious assessment of errors in movement, these areas. So now let's talk about the intracerebellar nuclei. So what you see here, they are the intracerebellar nuclei. So 4 masses of gray matter are lying deep in the white matter of the cerebellum on each side of the midline. So this is the midline, this is the cerebellum, and these are the 4 masses. And I previously called them the 3 masses, and now I will tell you how they are 3. Actually, these two, the intermediate ones, emboliform and globose, I will tell you. These are the grouped together. It's called as the given a common name of the interposed nuclei. So from lateral to medial, the names are the dentate nucleus. Here is a dentate nucleus. Then is the emboliform nucleus, and then is the globose nucleus, and the medial most is the vestigial nucleus. Here you see. So we can enlarge it for you, sure. Here you see the 4 nuclei on each side here. This is the dentate nucleus. Yes, this is the dentate. This is the emboliform nucleus and globose nucleus, collectively known as the intermediate nucleus or the interposed nucleus. And this is the medial most vestigial nucleus. So a mnemonic for remembering from lateral to medial side is: the doctors eat good food. So D for dentate, E for emboliform, G for globose, and F for vestigial. So dentate nucleus is the largest of the cerebellar nuclei. This one is the largest, right? And it has the shape of a crumpled bag with opening facing medially. You see again, it is a crumpled bag with the opening medially, right? And here inside is the white matter made up of efferent fibers, right? The interior of bag is filled with white matter made up of efferent fibers that leave the nucleus through the opening in to form a large part of the superior cerebellar peduncle. So next is the emboliform nucleus. Here this. It is located medial to the dentate nucleus, covering its hilus partially. Yes, here it is, right? Here you could see again. Here I can enlarge again. Here, this is emboliform. This is, this is the dentate, and this is its hilus, and here it is the emboliform nucleus. And then is the globose nucleus. This is the globose. It consists of one or more rounded cell groups that lie medial to the emboliform nucleus. Here it is lying, right? This is the your globose. Emboliform and globose nuclei are collectively called interposed nuclei. Then in the last, the medialmost vestigial nucleus. Here, this is the vestigial nucleus, which is lying close to the midline in the vermis. It lies in the vermis, right? It lies in the vermis. These two lie in the paravermis, and the dentate nucleus lies in the cerebellar lobe, in the cerebellar right lateral hemisphere. It's right. Again, I repeat, these two nuclei on each side lie. Vestigial nucleus lie in the vermis, and these two, your interposed nucleus lie in the paravermis, and this dentate nucleus lies in the cerebellar hemisphere, lateral part. Vestigial nucleus lies close to the midline in the vermis and near the roof of the fourth ventricle. So white matter, right? The amount of white matter is very less in the vermis, sparse. It's closely resemble the trunk and branches of a tree. Thus is called the arbor vitae. I told you already in this picture, and this is the arbor vitae, which is in Latin means the tree, little tree, the tree of life, tree of life. There is present a large amount of white matter in each cerebellar hemisphere. So white matter consists of three groups of fibers: intrinsic, afferent, efferent. Intrinsic fibers, they do not leave the cerebellum and serve to connect its different regions. Some interconnect folia of the cerebellar cortex and vermis ipsilaterally, while others connect the two cerebellar hemispheres together. Afferent fibers constitute the greater part of the white matter and proceed to the cerebellar cortex. These enter the contralateral cortex via middle cerebellar peduncles. Contralateral proprioceptive information and enters cerebellum via inferior cerebellar peduncle from spinal cord. And the detail of the fibers, what the different names of the fibers and where precisely they come from, that I will upload in a separate lecture, connections of the cerebellum, next lecture probably. Sure. So the efferent fibers constitute the output of the cerebellum and start as the axons of the Purkinje cells of the cerebellar cortex. Told already, the great majority of the Purkinje cell axons pass to and synapse with the neurons of the cerebellar nuclei. So the vestigial, globose, emboliform, and dentate. The axons of the neurons then leave the cerebellum. Fibers from the dentate, emboliform, and globose nuclei leave the cerebellum through superior cerebellar peduncle. Fibers from the vestigial nucleus leave through the inferior cerebellar peduncle. Right? I told you already this thing, that the detailed connections I will discuss in the next video. So it's very, very important, and some people say this is very complicated, but I've tried my level best to make it very simple. The cortical mechanism, cerebellar cortical mechanism. So what are they? So beautiful diagrams are here, and tables and flow charts, and please focus on that. Just it would be very clear. Sure, I'm very sure we have made it very conceivable. Some basic mechanisms are attributed to the cerebellar cortex. Climbing and mossy fibers form the two main lines of input to the cortex and are excitatory to the Purkinje cells. Here you see this, you know already that these three layers are of the cerebellar cortex, these three layers, right? Which is the molecular layer, Purkinje cell layer, and the granular layer. This is the white matter. And these fibers, the climbing fibers and the mossy fibers, right? These are excitatory to these Purkinje cells, which is, I will tell you later, is the main functional unit, right? Which is a Purkinje cell. So first of all, I will talk about the climbing fibers. See, you see, these are the climbing fibers. What are they? They are terminal fibers of cerebellar tracts. They are so called because they ascend through the cortex layers like a vine on a tree. Climbing, they'll ascend like a vine on the tree. Here you see, they go like this, like this. They ascend through the white matter and the layers of all the three layers of the cortex, and they reach this Purkinje cell dendroids. So they are ascending like a vine. They traverse the granular layer of the cortex and end in the molecular layer by dividing repeatedly. They divide repeatedly here. Each climbing fiber wraps around and makes a large number of synapses with the dendrites of a Purkinje cell. Here you could see. And that's a beautiful picture that will show you. As you see, these are the various numerous branches of the, terminals of the climbing fibers, which are making synapses with the Purkinje cells. A single Purkinje neuron makes synapse with only one climbing fiber, whereas one climbing fiber makes contact with 1 to 10 Purkinje neurons. A few side buds from climbing fiber synapse with the stellate cells and basket cells. So then is the mossy fibers. Here you see these are the mossy fibers. So what are they? These are the terminal fibers of all other cerebellar afferent tracts. So that was the thing. And they have numerous branches and produce a much more diffuse excitatory effect. Here you saw, this is the thing. Here I will discuss the forma glomerulus as well. I will talk subsequently very soon. They have numerous branches and produce a much more diffuse excitatory effect. And a single mossy fiber stimulate thousands of Purkinje cells via the granule cells, right? So this is the thing. Here they are coming, and here with the granule cells they form the glomerulus, cerebellar glomerulus, and in this way they stimulate thousands of Purkinje cells, a single mossy fiber. So then is very important functional unit of cerebellar cortex. What is that? So let us study that. Stellate, basket and Golgi cells are inhibitory interneurons. They limit the area of cortex, excite it, and influence the degree of Purkinje cell excitation produced by climbing and mossy fiber input. In this way, fluctuating inhibitory impulses are transmitted by the Purkinje cells to the intracerebellar nuclei, which then modify muscular activity through the motor control areas of the brainstem and cerebral cortex. So the Purkinje cells form the center of a functional unit of the cerebellar cortex. I will elaborate this slide, what is written. Deep cerebellar nuclei receive afferents via two sources. Afferents. There is the inhibitory axons from Purkinje cells of the overlying cortex, and the excitatory axons that are branches of the afferent climbing and mossy fibers that are passing into the overlying cortex. In this way, a certain specific sensory input to the cerebellum sends excitatory information to the nuclei, which after a short time receive cortical inhibitory information from the Purkinje cells. Efferent information from deep cerebellar nuclei exits the cerebellum and reaches the remainder of the brain and the spinal cord. So cerebellar cortical neurotransmitters. Excitatory climbing and mossy afferent fibers utilize glutamate as the excitatory neurotransmitter on the dendrites of the Purkinje cells. Other afferent fibers that reach the cortex release serotonin and norepinephrine at their endings that modify the action of the glutamate on Purkinje cells. Purkinje cells use GABA, gamma aminobutyric acid, as neurotransmitter to exert inhibitory action. So in the last, let me tell you what is a cerebellar glomerulus. Let's study that. Cerebellar glomerulus. It is a small interlaced mass of the nerve fibers in the granular layer of the cerebellar cortex. Here is the granular layer. Here it is the granule formed, this glomerulus formed. So parts of the glomerulus are, you can say, three: postsynaptic granule cell dendrites. Right, these are the granule cells, they're the dendrites. I will show you in a zoom form. And presynaptic Golgi cell axon terminals. These are the Golgi cell, their axons, presynaptic axon terminal, and the mossy fiber presynaptic terminals. Right, I show you these three things. This is a cerebellar glomerulus. Right, you see again. Yes, this is the a granule cell and its dendrites, and this is the Golgi cell, and this is its axons. Then they synapse over here, and this is whole thing is the glomerulus. And the third thing involved is the mossy fiber nerve terminal. Yes, this is the mossy fiber and its right nerve terminal. So this is the glomerulus formed, cerebellar glomerulus. So with this, I end the structure of the cerebellum. And in the next topic, I will discuss with you the connection of the cerebellum. I thank you very much and say for your cooperation, and please comment down below, and you please subscribe my channel for my support. And I say goodbye. Take care.